What we're going to do now is like doing the vision lab. And some of it, the first part we can do together. So, and again, if you've never done this, this is another thing that is really weird and cool. So this vision lab, special senses, vision. The first part of this lab is demonstrating that blind spot. I mentioned how on the back of the retina, there is that place where all of the axons leave, and where that place happens, there is no rod or cone. So there's no photoreceptor. So if this gray circle here is the back of your eye, the optic disc, that little spot there is a spot where there's no photoreceptors. That means if the image lands there, you have no sensory transduction. There's no information going to your brain about what is landing there. This is kind of showing, here's photoreceptors. This blind spot is where there's just axons. There's no photoreceptors. So what happens if you have an image that does land on the blind spot?
Your brain makes something up.
Yeah, your brain makes something up. So we can demonstrate this really, really easily, and it's fun. Let me turn this off. So in fact, we can even just do it on, normally I have it, you do it on a piece of paper in front of you, but you can actually do it on your screen in front of you even. I just put on this R and the L. This is a page from, this is a PDF of your lab manual, but you can just do it right now. If you shut your left eye, so you have your right eye open and you look at the R. So you keep staring at the R, but you notice the L. And then you move back and forth. When you're a distance, like maybe 2 to 3 times the width of the letters, you're probably going to notice something very bizarre happen to the L. Don't go side to side, go front and back. It's about the distance, and go kind of slow, because the little place where it happens is going to be, so start like, maybe the same distance between the letters, and then back up. And at one point, hopefully everybody's going to see the L. Something really weird happened to the L. Is it happening for everybody?
Yeah, for me.
It's supposed to disappear.
It disappears.
Yeah, it disappears. Is there anybody not seeing it disappear yet? So Sienna, are you using you're using your right eye to look at the R?
Yeah.
Okay, and just stare at the R, but notice the L, and get relatively close. You're going too fast. You need to kind of just slowly change your distance, and you'll find it. Slow. You're still going fast.
Oh yeah, I got it.
You got it?
Yeah.
So did everyone experience that? Right, so your brain is basically, doesn't know what's on the blind spot. Definitely doesn't know there's an L there. So it's like, it's white above, it's white below, it's white to the left, it's white to the right. What are the chances it's not just more white? So it just fills it in, makes it up, and in this case, it's wrong. And the L disappears. Stuff I would, rather than do it all right now, when you are doing this on your own, you can do more of these. This one here, you stare at the little square here and notice the spokes. Your brain does more fancy stuff than just fill in like, white. It can converge spokes. It can fill in, complete a big line. It's pretty cool. If the blind spot is too big, people who have damage that's bigger than just a small little thing, you can have what's called a scotoma. There was, what's his name? James Thurber. There was like a famous cartoonist who had one of these. And his brain just made up all these images constantly. He just basically had this weird cartoon reel running all the time in the side of his field of vision, because his brain just never knew what was there and kept trying to fit things in, and it kept shifting all the time. And that was just, but he actually took advantage of it to kind of do his art. So blind spot, that's the first part of this, is just notice it, record. I want you to actually measure the distance. Once the thing disappears, you can measure how far is it from your eye to the page. And it's also just designed to kind of slow you down and make sure you record your results. So that's part one of this, is just the blind spot. John?
This is kind of a frivolous question, but like in the case of the cartoonist, or in the case of different people having that same, what was it called? Scotitis or something?
Scotoma.
Scotoma. Would somebody with like a greater overall, I don't know, a bigger imagination, like end up seeing more and have more interesting things happening when the brain can't actually process or doesn't have enough information to fill in a blind spot?
So that,
does that make sense?
Yeah, it does make sense. And I don't know. I mean, I do know that there are some stereotypical kind of things that come up just because of the structure of your brain and your visual cortex. Like people who studied like hallucinogens and things like that have found very typical kinds of patterns that people see, that is depending more on kind of the basic wiring and layout of the neurons in your visual cortex versus what your imagination is. That being said, I'm sure your learning and imagination play a big role as well. Right, so I think I'd say the answer is probably a mix of yes and no.
That makes sense.
Yeah. Let's see.
So you want us actually measuring on that one? Because all it says is, why does this work? So, but you want us actually measuring?
So it says here, have your laboratory partner, or so it's either somebody there or you can try to do it yourself, measure, record the centimeters, the distance at which this, so just read this carefully and follow the instructions there. So that's what I would say. So there is, you have the lab and the directions. So just follow that one there. So after images. So this happens when you stimulate the photoreceptors strongly. There's two things that happen. One is they can keep responding for a little bit after you've stimulated them, kind of like there's a loud sound and your ears keep ringing for a while or something. So like there's a bright thing and you keep seeing a bright thing for a little while. That's like a positive after image. That you can see just by like shining a light in your eye. Where is it? In fact, let me find my light. Back. Up here. So you can shine a light in your eye, turn it off. Spoiler alert, I see a big red, yellow dot. But now it's actually changing. And what happens is if you get your photoreceptors like overexcited, they can kind of bleach out, and basically it takes them a while to come back online. So then you have what's called a negative after image, like the classic green flash, like people say they see like on a sunset or something. You're seeing this kind of red, really bright, and you basically kind of tire out the red photoreceptors. And so when the light goes away, there's like no red, and actually the absence of red in your eyes looks like green. So the negative after image of kind of overstimulating the red receptors is everything looks kind of green, because you don't have any ability to detect red anymore. Normally red and green kind of balance each other out in this world of color illumination. So one of the things I have for you to do, so I have here, stare into the bright pen light bulb. You could just stare at a bright light of any sort, and just describe what you see. But then I have another thing for you to do that is actually really cool. So here on part 2, demonstrating after images, this says do at home, record in your lab notebook as well. It says also do this cool YouTube example and record your observation. We can actually do, so this is actually kind of a cool thing. So this is a picture of a bee, but with all the colors inverted. So, and again, you'll have to just trust that these are the opposites of the colors. And so if you're staring at this for a long time, the photoreceptors that are responding to the colors in the video in front of you are getting tired. And when this thing goes away, what's going to happen is your visual color experience is going to be kind of the opposite of what you were seeing, because the photoreceptors that have been watching this have gotten overstimulated. So in fact, we can just do it. It's fun. It's cool. So you're going to stare at this dot for 30 seconds once it comes on. And as soon as, and you're going to see, we're going to go to a black and white picture of a bee. But you're going to see for a moment, it's not going to. So stare at that purple dot in the middle. It's important that you maintain fixation, because you want the same cones to keep seeing the fuchsia or the green or the blue, so they get kind of overstimulated and kind of lose their ability to respond after a while as strongly. So just keep staring exactly at that purple dot without shifting your vision whatsoever. So did everybody see, what did the bee look like when it first flipped onto the black and white?
Yellow.
It looked yellow, but it wasn't. Like if I go back here, it went straight from this to black and white. There is no yellow in this video.
That's trippy.
That is cool.
Right, so that's an example of that negative after image. So yeah, it's pretty cool. But that's kind of that, again, most people have heard of like the green flash on the sunset. It's the same thing. The red disappears and then there's a green after image left where there's no more red light. It's kind of the same thing, except instead of a inverse B, it's a red sun. So do that. But in your lab book, kind of record your results. The whole point is you describe what you see and kind of talk about what you think is going on.
So just to be clear on that, by staring at that kind of purplish dot and focusing on it, you were kind of overloading all the receptors that could take in that color. And so when it switched to black and white, like the opposite colors, or I don't know if opposite's the right word, those kind of overcompensated at that point.
Yeah, so like red and, I mean, I wonder if I have a picture of like one of the classics is like the red and green bird. Let me go in like images, Google, red, green bird. No, this isn't quite what I meant. Like if you stare at a red just picture of a bird for a while and then the bird disappears, for a moment, you're just going to see a green bird. Or if you look at a green bird for a while and then it disappears, you're going to see a ghostly red bird for a moment. Just because normally the red and the green kind of balance, but if you've been staring just at the red bird, the red receptors are getting tired. So when that bird disappears, then the greens have a little extra dominance that they normally wouldn't have. Right? Normally the red and the green are balancing each other out. But if one of them is getting tired, then these guys have a little dominance for a moment until these guys come back online. If that makes sense. Yeah, this, I didn't mean to have these pictures of the birds. All right. Color blindness. So color blindness, this is, I think it was Nikki mentioned, this has to do with a genetic problem where you just are lacking the gene to make cones that respond to a particular color. This is what we call a sex-linked trait. You know, why, what do we mean by a sex-linked trait?
It's comparing male versus female, right? Like sex
No,
meaning it's more dominant in either a male or a female.
no, it's not that. It is more, it's much more likely to happen in a guy than a woman.
Is it on the Y chromosome?
Almost.
I think it's on the X chromosome, right?
Yeah, if it was on the Y chromosome, then like you can't have anything on the Y chromosome that's important to just be a human in general, because right, everything you need to be a human's got to be like on an X. The Y just has like some maleness stuff on it. So the code to make these opsins, to make the things you need to make these color cones are on the X chromosome. And if you're a woman, you've got 2 X's. So if you have one bad copy, you're probably okay because you got 2 X's, you brought a backup copy, right? You have 2 alleles of everything. So it's pretty rare for a woman to be colorblind because you'd have to have 2 bad hits of a damaged gene to make a particular cone. But for a guy, you only have 1 X chromosome, right? You're an X and a Y for your sex chromosome pair. And so if you have 1 bad copy, that's it. You're colorblind. So that's why, you know, in this class, I have over the years had lots of guys who are colorblind when we do these colorblind tests. I have never ever seen colorblind women do exist, but I've never met one. Again, you'd have to be particularly unlucky to be a colorblind woman. You know, the way that you can tell if someone's colorblind is with these special plates. They're called Ishihara test plates. And I'll show you how it works. This is actually going to take you a little bit of time, but it's pretty cool. This This guy, some of these guys, I feel bad that I kind of make fun of them, but they kind of crack me up. This guy cracks me up. But he's providing a very valuable service here, demonstrating the color blindness test. And he actually goes through it so you can actually record your own results. So there are a sequence of these. And
so are you colorblind? Well, in this video, I'm going to be walking you through the Ishihara colorblind test so that you can figure out if you were colorblind or not. Let's take a look. Hello and welcome. This is Dr. Alan here from the Dr. Eye Health Show, helping you learn all about the eyes and vision. And I'm excited today because we're going to walk through an Ishihara colorblind test. This is the most famous colorblind test that's out there. This is the one that you've probably seen with little colored circles on it. But this is going to be cool because we're going to, one, show you really.
All right, so you got to admire how his like eyes match his shirt and everything. He's obviously somebody, he kind of really kind of set things up for his video. So like these are these plates, and you look at them and, people with normal color vision see like a 7 and a 4, in these dots among all the other dots. But these are designed so that a lot of these dots vary by amounts of red or amounts of green. So if people have different kinds of color blindness, sometimes some of these dots actually don't look any different from the other dots if you're colorblind, even though they do look different if you have normal color vision. So what he does is he goes through here and you take a look. This one is like the control one. This one is designed so no matter what kind of vision you have, you're going to see a 12. All the orange dots are just kind of bright orange. All the background dots are the same kind of blue. But once we get into here, if you see an 8 versus a 5 and stuff, some of these look like random. Some of them are kind of cool in that they look random unless you're colorblind. You only see a pattern if you are colorblind. But so you're going to go through along with, what's his name? Alan? Alan will guide you through the different pages.
70. On the next page over, you should see a 7.
Right. He's going to take you in your lab notebook. I want you to write down, the plates have a different number. He'll tell you what number the plate is. There's plate 4, plate 5. The numbers are so freaking dinky. I always bum that they're so small. I think he tells you what the numbers are as you go through them. And you're going to describe what you see and what it means. Hopefully for most of you, it's going to be like, that's what I should see for normal color vision. Or it might be that you're colorblind, in which case you record that too. But so this is just going to take you 7 minutes and you're going to go through with your guide and he's going to just take you page by page. You get to, yeah.
You've gotten correct so far. If you've had 10 or more of this.
And he's got really bright teeth. I don't know why. No, again, I really appreciate that he made this thing. And I think this will be useful. If you haven't done this, it's useful to see how you do it. And it also gives you a sense of what it means to be colorblind. Because even if you do see this stuff, realizing that someone with color blindness doesn't see it, and you get to like think about what would that be like. It's also a useful exercise to actually get a bit more of a intuitive sense of what would the world look like if it was colorblind. You'd still see everything as objects, but again, like it might be browns and greens that look the same to them are obviously different colors to you and me or something. Yeah, my uncle, yeah, it was funny. I had my uncle is pretty deeply colorblind. I remember like, how do you know like when to go or stop at a stoplight? And he's like, I'd never thought that, oh, the red's on the top and the green's always on the bottom. They didn't have to care about the color because there's other clues you can use to know whether it's time ready to stop or go. But anyway, so any questions about the Ishihara color test? So you're just going to sit with this dude for 7 minutes, record what you see, and interpret the results. Interpret what does it mean in terms of your color vision. And all right, that's it for Mary's in his groovy glasses. All right. Visual acuity. All right, so we now have to give a little background on how the lens focuses the image on the retina. So the next part's visual acuity, astigmatism, near point of accommodation. Those are all related to how your eye focuses images and gets a clear focused image on the retina. So I need to do a little bit of background information to talk about how that works. So let's do that. All right, so I mentioned that there's a lens in your eyeball that helps focus. We need to talk a little bit more about lenses and optics. A lens that bulges on the side is called convex. This is the kind of lens that's inside your eye. It's a convex lens. A convex lens converges light rays. If light rays come in, it will bend them inward. We call that converging. If you change how, hold on. Okay, I think is my voice sound right? Okay, yeah, I had a weird Bluetooth hijacking where my microphone got hijacked. I think I'm back. All right, so those little muscles, those ciliary muscles, those muscles inside your eye, they're more smooth muscles that are being operated by your autonomic motor. They can change how bulgy this convex lens is. So if I make the lens more bulged, then it will bend the light even more. So I can change how much the light converges by changing the how convex the lens is. The more convex, the more bulged it is, the more it will bend the light. The closer in this point of convergence, this kind of focal point is going to be. So this is how we're going to make the image land on the retina. We can adjust the amount of bending of convex on the lens, and we can bring the image into focus wherever the retina is. While we're here, we should also talk about concave lens. Concave lenses are going to be useful for corrective lenses. You need to know them so you know how glasses work. They're not in your eyeball, but they're going to be useful for understanding glasses. These do the opposite. These will diverge the light rays. The way I remember when I was first learning this, if you get confused about what is convex, concave, I was like thought about like if you're a bear, it's kind of, it's October, it's winter time, it's eating all your salmon. Now it's time to try to go and hibernate. You got to find like a little cave to go into, right? This looks like kind of the entrance to a cave versus there's no place to go and sleep on a convex lens. If that's interesting, if that's useful or not. I don't know if you saw that bear that's been in the news, that big fat bear up in Alaska eating all the salmon. He's pretty cool. All right. So this being said, convex lens, which will converge the light rays so we can bring this focal point either further out or closer in, depending on how much we bend the lens like this. So what I'm going to do now is draw an eyeball again, but now we'll look a little more rigorously at how the light is focusing in there and how the lens affects that. So we are going to start with what is called the emmetropic eye. This is just the fancy word for normal vision. This is like somebody that's 20/20, somebody who does not need to wear glasses. This is if the eye is just kind of working kind of quote unquote normal. Right. And I was that the word normal is always a little weird because, it's like how many people actually need to wear glasses? A huge part of the population. So like saying that doesn't mean they're not normal. It's actually pretty normal to wear glasses as well. But we're going to call this kind of normal vision. Eyeball. There's going to be the lens. There's the retina. Retina is kind of like the film, so to speak. And then there's going to be some object that we're looking at. So in my first picture, I'm going to draw something that's at a far distance. 20 feet or further away is the typical distance for being considered far distance. And let's put I usually put a deer there, or elk. This can be that elk that they just managed to get like the tire off of him, and he'd been wearing a tire for like 2 years. I don't know if you just saw that in the news. They like shot him down with a dart. They had to cut his antlers off, which was a little sad because they couldn't get through the steel belts in the tires. So they had to like cut his antlers off to get it over his head. But here he is. He's there 20 feet away. The light rays from this guy are coming into your eye. The lens, in fact, let me do this less. The light rays, dang nabbit. The light rays are coming in. At 20 feet and beyond, there is this basic assumption in kind of this optics that the light rays are coming in parallel. So these light rays are coming in parallel. The lens, when you are just having your eye relaxed, the lens has just the right amount of curvature so that it bends the light rays so they land right on the retina, and this elk with the tire is in focus. So the lens is relaxed. And if I look at things 20 feet and beyond, it has just the right amount of curvature that the things will converge just right on the retina, and anything I look at in the distance will look like it's in focus. And again, you're not using any muscles. This is kind of the natural curvature of the lens when the muscles are relaxed. If you look at something closer, then you have this kind of diverging light rays. So let's look at, I don't know, bunny rabbit. This bunny rabbit's up closer. And the light rays from the bunny rabbit are no longer this parallel assumption because he's not far away. If you're closer, then again, I'm kind of playing fast and loose with my ray diagrams, but I think this will give you a good sense of what's going on here. So the light rays are diverging from this thing that's close. What does my lens need to do? Because right now it means that they are going to focus too far behind. So what does my lens need to do to bring this rabbit into focus?
Bulge more.
Needs to bulge more, exactly. So the lens will get more bulgy, which means now that the rays will bend more, and now the rabbit's in focus. So that process of having the lens bulge more to bring the rabbit into focus or anything close into focus is called accommodation. So in fact, I will use I keep forgetting I have the ability to type. So accommodation is the ability to adjust the lens. So as things get closer, you can bend the light even more so you can keep it landing on the retina and keep it in focus. You know, at some point, something's going to get close enough. You know, let's say, oh, I need another color. Let's say this, if something is close enough and diverging, you're going to overwhelm. There's a limit to how much the lens can actually bulge. So that means there's a limit to how close something can get, and you can still bring it into focus. So that limit, the nearest something can get, and you can still focus on it, given the limits of your lens, is called the near point of accommodation. So the near point of accommodation, the closest you can be to the eye and still have the capacity to adjust the lens, to accommodate, to bring the thing into focus. If it gets closer than that, no matter how hard you try, it's just going to be blurry. um, how close can you usually bring things and still, like, in fact, we should ask a younger person to like just bring something closer. How close can you get before things get blurry? Like use one eye.
Within a couple inches, maybe.
Couple of inches, yeah. So then like 6-8 centimeters usually. What happens as people get in 40s, 50s beyond, the lens becomes less flexible. And the lens loses its ability to change its curvature as much. So what is that going to cause if the lens has less of an ability to curve?
Farsightedness.
No. We're not getting into nearer farsightedness yet. Right now, we're talking about near point.
Blurry vision up close.
Yeah, it's going to mean, right? What's going to happen to the near point of accommodation as you get older and the lens loses the ability to accommodate as much, to bend as much?
Gonna get further out.
Get things further out.
Going to get further out. Exactly. The near point of accommodation, the closest you can bring something without it being blurry, is going to be further and further away. Yeah, I can say when I got into my early 50s, it was very disconcerting, and at this point, like I carry readers around. These are basically things to adjust to let me focus on things really close. If you've probably seen older people trying to read the menu in a restaurant and holding it further and further away. That's because their near point of accommodation is retreating. One of the things that you'll have, in fact, I can just show you right now. Kind of teaser alert. This is the data that we have from another class of near point versus age. The older you get, the further it goes. That's just the nature of, there's lots, we're going to see as we go through this semester, there's going to be more and more and more bad news about your vision, your hearing, all sorts of things. Pretty much, I mean, the reality is most of you guys are already over the hill, even for super peak athletic performance. By the time you even get into your early 20s, your reflexes aren't what they were when you were like a teenager. So, the good news is that you can compensate in different ways and be more clever or just do different things, but or technology, like get some cheaters.
Did you have to say over the hill?
Did I accidentally say that?
I'm joking, but I think I heard that. It's true.
No, I mean, so there is actually evidence even in the brain imaging where they have people do the same task. It might involve working memory and this and that. And externally, if you just look at performance on the task, it looks pretty similar. But if you're looking at the brain regions being recruited and being used, it's actually different in younger people versus older people. It does seem that, certain things, what's the right word? Like you can lose some performance on certain circuits and this and that, but it doesn't matter in the big picture of what you can do because you can bring other things online or do kind of change strategies. So the idea of getting older sometimes is not so much like you get worse, but you do things differently in your nervous system. But some core things like reflexes just get worse. But I need to keep talking because we are getting down to 15 minutes. Okay, so accommodation. Accommodation is this idea that you adjust the curvature of the lens to bring things into focus as it gets closer. The near point of accommodation is the closest you can bring it without it being blurry. You know, we are going to have you guys do that in your lab, measure your near point, and then, you know, print out that graph I just showed you and kind of talk about the bigger picture of what happens as you get older. Now what we're going to do is talk about a couple of the things that can go wrong with your vision and visual acuity. So one thing that can happen is astigmatism. So astigmatism is when there is some asymmetry, spherical asymmetry. In either the cornea or the lens. So instead of being perfectly spherical, meaning like doesn't matter what angle you're coming in from, it's all the same, it's more like a football, right? One side of the lens has some amount of curvature, but coming in horizontally, it's actually got a bit of a different curvature. So light coming in from one angle is going to, or one axis is going to be bent differently than light coming in on another axis. To test this, we have this thing, it looks kind of like a big asterisk. This kind of thing right here. With the 12 and the 6, the 8 and the, you know, I'll have one for you to print out, a prettier one. But the basic idea is, even though the actual chart is completely symmetrical, most people have astigmatism are going to see that one axis looks either brighter or darker or blurrier or thinner or thicker than another axis because it's being refracted slightly differently than the lines coming in on a different axis. So that is going to be like the test for astigmatism is, in fact, if we go to, here it is, astigmatism chart, JPEG. So you
We
can
can't see anything, just so you know.
Sorry. Oh my God, you're so sorry about that. So here's this test for astigmatism, you know, which you're going to do at home, record in your lab notebook. You could actually probably just put it on your screen or you could print it out. The basic idea is you do one eye and then the other, and you look to see if all of the axes are the same or not. Like for me, they're not. Like 12 and 6 are definitely much more clear than 1 and 7. So that is astigmatism. Astigmatism is this asymmetry. The lines on one axis are going to be processed a little different than lines from a different axis. So that's something you'll be able to look at home. You know, the other thing that we talk about is like nearsighted and farsighted. So let's talk about what that means. Nearsighted is myopia. Farsighted, hyperopia. As opposed, versus, the emmetropic eye. The normal eye. So we're going to talk now about eyes that are not emmetropic, that are going to be farsighted or nearsighted. And one way to think about it is like of an elongated or shortened eyeball. So here's one option. So this is supposed to be this kind of the eyeball I was drawing, but now it's kind of elongated. This is going to be myopia. And now I'm going to look at this elk over here. Let's tie her around his neck. Actually, not anymore, but I'll put it there just for nostalgia's sake. And the light rays are coming in. Like I said, light rays from, let's say this is 20 feet away. Light rays coming in, they're like parallel. And then the lens is the normal lens, which is designed is, kind of got the right amount of curvature to normally make this thing at 20 feet away land on the retina. But because the eyeball is elongated, normally the retina would be there, but it's not because the eyeball is elongated. So it's elongated eyeball. So by the time the image gets to the retina, it's blurry. So this elk is blurry. I'm not even sure this person's going to even notice that there's a tire around his neck. Is there anything that this person can do to get this elk into focus?
Wear bifocals?
Let's not use glasses yet.
What about squinting?
Squinting.
So that's something different. So squinting can make a difference by changing the aperture. You actually change the depth of field, but we're not going to get into that right now. Let's think about the lens.
Relax the lens so it's less bulgy.
Right. So that theoretically, if you could make it even less bulgy, that would help. But the problem is it's already the least bulge it can be. The muscles only make it more bulgy. So basically, somebody who is myopic, nearsighted, there's nothing they can do, at least using their lens, to bring this thing into focus. When you look at things that are closer, like that bunny rabbit that was closer up, where the light was diverging to start with, that means that it's going to be landing further back because it's already diverging to begin with. So you actually can focus on the bunny, the close-up thing. That's why this person is nearsighted, because things that are close, that have diverging rays to start with, that's going to be all right. They're going to be able to adjust to get that into focus. But things that are 20 feet or more away, there's nothing they can do, short of glasses. What would you do to help this person out? What kind of glasses, what kind of lens, corrective lens would you use?
Concave.
Exactly, concave. So if I take a concave lens, so that the rays that are coming in initially diverge a little because of the concave of the lens. Then when this lens is at its normal, just relaxed thing, it's going to be just right. So you basically find the concave curvature of your convex lens to just make it so when the eyeball is relaxed, the image lands exactly where it's supposed to be. And now everything is just the same as the emmetropic eye. Wait, yeah. So does that make sense? Okay, so, oh my God, the time. Let's continue here. Let's say we have an eyeball that's short. It's going to be very different. They're going to look at this elk here. Now they're going to, the place where the retina normally would be further back, because now this is a shortened eyeball. But because the lens can get bulgier, we can bring this deer into focus, this elk into focus. If we accommodate, we can make the lens bulgier, and we can bend the rays more. And so we can see this elk at a distance. But the problem is we've just used our accommodating power already. Normally, you can see something at a distance, just letting your eye be relaxed. This person's having to work to see this distant elk. And they're using up all their accommodating power just to deal with bringing it into focus on this shortened eyeball. So there's no accommodation power left if we start looking at things closer. So while this person can look at things that are far, they can't see things that are coming up close because they've already used up their accommodation power just trying to adjust to far distance things. So the farsighted or hyperopic person, they can see things far away without glasses, although their eye has to work in ways it normally wouldn't. Normally, you would not have to tense those muscles and bulge out that lens. But they can't see things that are getting closer because there's no more accommodation power left. So that is the basic idea. For this person, you're going to obviously use a convex lens as a corrective lens. So the light rays coming in get pre-bent a bit. So then they will land on the retina just with the relaxed lens. You can tell if you just take somebody's glasses, right? This is a magnifying glass. This one actually makes things look smaller. If you take someone's glasses and put them over a piece of print and see whether or not the letters look dinky or whether they look magnified, you know whether they have glasses for farsighted or nearsighted.
I have a question.
Uh-huh.
Is it always the depth of the eye that causes these problems, or can it also be the nature of the lens?
I think it can be other things as well. This is the way I usually kind of understand it, but I think there can be other things that lead to the same basic effect. Right. So it could be the lens is just naturally, because obviously it's a mismatch between the curvature of the relaxed lens and the distance to the retina. So any of those moving parts could result in this thing. So how do you test if somebody has emmetropic eye or is like nearsighted or something?
Through that like eye chart.
Yeah, the eye chart. Exactly. Its official name is the Snellen Eye Chart. the Snellen Eye Chart, which I'll share with you. It's this kind of thing, which we have ones for you to print out. And they have letters at different layers, different little to big. This one is kind of designed to print at home and do. It's not as nice as the ones we would have used in lab or you'd use in an actual, and actually some of the stuff in the actual eye exams now use more projected things and stuff. But the basic idea in a classic eye chart, you one eye at a time, you look at the chart here, you stand 20 feet away. There's that 20 feet again. It actually is important that you're standing 20 feet away. And then, E, L, T, and smaller and smaller letters. And each level has it'll say, this one is usually 2020. This is 2015. This is 2040. This might be 2080. So you basically have to read the different things. Somebody else has to be checking to see how you're doing. And you find what is the line that you can read correctly, the smallest line. If you make a mistake, you can't count that. You have to pick what is the last line you could read without making any mistake. And let's say it was this line, the line that said 2040. You could read those letters without a mistake, but you made a mistake on the next one. So 2040, what it means is this is you. You are standing 20 feet away. This is an emmetropic vision. You're standing 20 feet away and you see with the same visual acuity if a normal eye was standing 40 feet away. So a normal vision person could be standing 40 feet from something and see as clearly as you see it at 20 feet away. What if I could only read this, 2080? What would that mean?
That a normal person would be able to see that from 80 feet away.
Yeah, so that's even worse vision for you. That means you're standing 20 feet away and you see what a normal person vision would see if he was standing 80 feet away from it. Our chart doesn't have this. Most of the more professional charts do. It is possible to get like 2015. Especially if you're reading the smallest one at the very bottom. What would that mean?
That you can read from 20 feet what an emmetropic eye, what a normal person with good vision with 20/20 could read from 15 feet away.
Yeah, so normal vision would actually have to be closer than you to see as good as you. You have better than normal vision. And then we should just, for completeness, what does it mean to be 20/20?
Perfect vision.
What's that?
Is it like supposed to be perfect vision, the ideal eyesight?
But just thinking about what the 20 means, 20.
20 feet at 20 feet.
Yeah, so at 20 feet, you see what a normal vision person would see at 20 feet. Oh, you're normal. I mean, you can be better than normal. 2015, there's a lot of people actually that are better than 20/20. So 2020, I wouldn't say is perfect. 2020 is more normal, I would say. But who knows what is perfect? You know, perfect is in the eye of the beholder. But so this is something I would like you all to do is, you know, this chart is designed to be able to do at home and do from 6 feet instead of from 20 feet. So it's, you can do it in a bedroom. And you're going to obviously need somebody to help you out. But the basic idea is find out what are the letters you can read and what does that say. And I recommend do it with glasses and without glasses. If your glasses are prescribed well, your glasses should bring you into 2020. And it's kind of fun to see that. And you do it one eye at a time. So that will be part of your lab. So near point. This oh my God. So binocular vision. You should, you know, I realize we're at 12:34. I am going to keep talking just for like about 3 minutes. And if you have to dip, this will be recorded. But I will just, for completeness, mention binocular vision. Like the fact that we have 2 eyes, right? Why, what do we gain from having 2 eyes?
Depth of field.
Yeah, a big part is depth of field. Like one eye or the other eye alone, if you just look at the world with one eye, everything looks way flatter. It looks different. So part of your, you know, just follow the thing in the lab manual. It talks about trying to put a pencil in a test tube, but you can put it in any kind of small thing. You basically just need to have somebody holding something, and then you try to quickly get a pencil into it and see how well you do with both eyes open versus one eye. And it's a pretty fun thing. It's like thinking about how much 2 eyes helps you. It's pretty amazing. It's also important to realize that there's more than just binocular vision to let you know how depth is playing out, right? There's people with only one eye that drive safely. What other things do we have in the world to let us know depth?
The size of things.
Yeah, perspective, right? Just it was something that revolutionized the art world like hundreds and hundreds of years ago. Just the farther things are out in the distance, the smaller they look. So like this picture here, this is to kind of show you how much you use size clues to figure out like what. In fact, here, I'll do this. I can do this because it will be cool. I'm going to draw, I'm going to make a line, maybe a box. What does it look like with these 3 different cars?
An optical illusion.
But what is your experience? What do you see?
That the one all the way in front is much, much bigger, and the one
This
closest
one
to
in
it-
the front looks way bigger than the one in the. So if I draw a line here, can I move it? Oh fuck! I want to. I know what I'm gonna maybe. Here, this is the size. This is actually about the size of the license plate. The license plate. The license plate. Each of these cars. This is about the height of the wheel well, a little smaller. Height of the wheel well, a little smaller. The height of the wheel well, a little smaller. Each of these. This is just a Photoshop cut and paste. 3 identical cars. They're not even kind of sort of same. They are identical cut and paste of the same car. So the reason, you'll have it on your screen. This is just in your lab in the module, just to let you realize that just the relative size of things is messing with you and giving you information about depth. It's not just stereoscopic vision. And how things move as well. Like, as you move around, some things move relative to others. Things in the foreground are going to move more or less than things in the foreground. So there's more than just binocular vision, but do the thing where you're testing 2 eyes versus 1 eye for dunking the pencil, but then also be aware that there's more to distance and more to like understanding depth than just that.